question 1 is about carbohydrates or polysaccharides, and we are asked to describe two differences between the structure of a cellulose molecule and a glycogen molecule. So just to recap, glycogen is an energy storage carbohydrate found in animals, whereas cellulose is a structural carbohydrate found in plant cell walls. There are a number of key differences between the structure of glycogen and cellulose, as you can see from this diagram here, as well as some other differences which aren't obvious in these diagrams. The first key difference between the structure of a cellulose molecule and a glycogen molecule is the monomer which makes them up, so the single unit which makes them up. The monomer for glycogen is alpha glucose, whereas the monomer for a cellulose molecule is beta glucose. And the difference between these two monomers, as shown in this diagram, is that the hydrogen is above at carbon 1 in alpha glucose and below at carbon 1 in beta glucose. So this is one key structural difference between these two molecules. Secondly, as you can see from the diagram shown of glycogen and cellulose molecules, the structure of glycogen is highly branched, whereas the structure of cellulose is made up of these long, straight chains. And this is important for the roles that they play as carbohydrates. Because glycogen is highly branched, it means it can be readily hydrolyzed to release lots and lots of glucose at once. This glucose can then be used in aerobic respiration to produce ATP. Cellulose, on the other hand, is structural in its role. It therefore needs to be strong and forms structures called microfibrils, which contribute to the strength of the molecule. So another key difference is that cellulose is made up of straight chains, whereas glycogen is branched. In addition to these two structural differences between a cellulose molecule and a glycogen molecule, you would also achieve the marks for stating that cellulose molecule has straight chains, whereas glycogen is coiled, or for stating that glycogen has 1,4 and 1,6 glycosidic bonds, which leads to the branching of the molecule, whereas cellulose has only 1,4 glycosidic bonds, and that is why the chains remain straight. For the next part of the question, we are asked to describe and explain two features of starch that make it a good storage molecule. So let's just recap. Starch is an energy storage carbohydrate found in plants. It has this branched structure and is made up of alpha glucose monomers. Now, very similar to glycogen, the energy storage carbohydrate found in animals, starch is highly branched, and this means that it can be readily hydrolyzed to release alpha glucose molecules used in aerobic respiration. The fact that starch is highly branched also makes it compact. And as we know, a compact molecule, especially an energy storage molecule, can store lots of energy in a small area, which is ideal when a plant is trying to store lots and lots of energy. Now, we've written enough and provided enough responses to achieve 2 marks. However, there is another way that you might have answered this question, and that is by stating that starch is insoluble in water. Okay, so it does not dissolve in water. What this means is that it doesn't affect the water potential inside cells, and therefore doesn't cause water to move from an area of high to low water potential by osmosis. And finally, for 1 mark, we're asked to tick the box that identifies the test which would be used to show the presence of starch. Now, the acid hydrolysis test is just another name for the non-reducing sugar test, so testing for sucrose. There's no direct test for sucrose. So what we first have to do is hydrolyze the glycosidic bond holding sucrose and fructose together by adding dilute hydrochloric acid and boiling it. Then what we do is neutralize the acid and then test the sample for a reducing sugar. However, that is not the appropriate test for starch, so we're not going to tick that box. Equally, the Benedict's test is testing for the presence of reducing sugar, for example, glucose. However, again, we're looking for a way of testing for starch, so this wouldn't be the appropriate test. The emulsion test is the test that we use to test for the presence of lipids in a solution. The way that we do this is place our sample into a test tube. We add ethanol or alcohol and give it a good shake. We then add distilled water to the test tube, and if we get a milky or cloudy emulsion, then lipid is present. However, starch is not a lipid, so this would not be the appropriate test. So by the process of elimination, we know that the correct answer is the iodine or the potassium iodide test. For this test, all you need to do is add iodine to your sample, and if it turns a dark blue-black colour, then starch is present. So this is the correct answer. The next question is a magnification calculation for which you're going to need your OMN triangle. O is the observed length of the structure, so measured with your ruler in millimetres. M is the magnification of the image, and N is the natural length of the structure. And the majority of the cases, this is going to be in micrometers. So we've been asked to calculate the actual diameter of the starch grain between points A and B. So we're looking to find N from our formula. So that makes our formula N equals O over M. In order to get our observed length of the structure, what we need to do is take our ruler and measure between points A and B in millimetres. So this distance here, which is 20 millimetres. We then divide that by our magnification, which is provided in the question, so 500. If we pop that in the calculator, we get 0.04. But importantly, this is still in millimetres, but we're asked for our answer in micrometers. To convert from millimetres to micrometers, we need to times by 1000, making our final answer 40 micrometers. Now you may have converted to micrometers at this point here, and dividing 20,000 micrometers by 500 will have also given you the answer 40 micrometers. We're asked to look at figure 1 and determine what type of microscope was used to obtain the image. And we need to provide evidence for the microscope that we have chosen. In AS Biology, we've learnt about 3 different types of microscopes. Firstly, we learnt about optical microscopes or light microscopes, which use light to form an image. This type of microscope has the lowest resolution of all of the microscopes and also the lowest magnification. This is the type of microscope we use in class. In addition, we learn about 2 different types of electron microscopes. The first type is a transmission electron microscope, or a TEM, which sends a beam of electrons through a specimen. And this produces an image because denser parts of the specimen, the organelles for example, absorb the electrons and the cytoplasm doesn't. So that allows us to produce an image of the structure inside of a cell. The second type of electron microscope that we've learned about is the scanning electron microscope, or an SEM. And SEMs scan a beam of electrons across the surface of the specimen. This results in a 3D image. So all of the different types of microscopes that we've learnt about have got their own relative advantages and disadvantages, but if we have a look at the image here in front of us, we can see that it has to have been produced by a scanning electron microscope because it is three-dimensional. This next question is about DNA, and we're asked to count how many nucleotides are shown in figure 1, which is part of a DNA molecule. Now, as we know, a nucleotide is the monomer, so the single unit, which makes up DNA. And as shown in this diagram here, a DNA nucleotide is made up of three key components: a phosphate group, a deoxyribose sugar, and a nitrogenous base. So shown in this diagram, we have 1, 2, 3, 4, 5, 6, 7, 8 nucleotides. For part 2, we're asked to name the type of bond labelled X in figure 1, and as you can see, the bond labelled X is present in the sugar phosphate part of the DNA molecule. So this is called the phosphodiester bond and joins nucleotides together between the deoxyribose sugar and the phosphate group. Just a little bit of extra revision. Here we also have hydrogen bonds present in DNA between the nitrogenous bases. 2 hydrogen bonds form between adenine and thymine bases, and 3 between cytosine and guanine bases. So this is another type of bond that you could get a question about when asked about the structure of DNA. A question about DNA replication now. The enzymes DNA helicase and DNA polymerase are both involved in DNA replication, and what we're asked to do is describe the function of each of these enzymes. So what is their role in DNA replication? So here we have a section of a DNA molecule which is about to undergo DNA replication. DNA helicase is an enzyme which breaks the hydrogen bonds holding the nitrogenous bases together, causing the 2 strands to separate, as shown here. Free nucleotides from the cytoplasm will then be attracted to the complementary base pairs on the exposed strands. And once these new nucleotides have been attracted to their complementary base pairs, they are joined together by the enzyme DNA polymerase. So DNA polymerase's job is to join the new nucleotides to form the new strand and build up the new sugar phosphate backbone, as we can see here. And it does this by forming the phosphodiester bonds that we discussed in the previous question. For the next part of the question, we're asked to contrast the structures of ATP and a nucleotide found in DNA to give 2 differences. So here is that diagram of a DNA nucleotide again. We can see the phosphate group, the deoxyribose sugar, and the nitrogenous base. And here is the structure of an ATP molecule. Instead of just having 1 phosphate group, ATP has 3 phosphate groups. And whilst ATP does have a 5-carbon or pentose sugar, instead of having deoxyribose like a DNA nucleotide, the pentose sugar present in ATP is ribose. We've seen ribose before because in an RNA nucleotide, ribose is present. We also have a nitrogenous base, but in ATP, the nitrogenous base is always adenine. We'll never see thymine, guanine, or cytosine. So the key differences between a DNA nucleotide and ATP in terms of their structure are that ATP has ribose sugar and a DNA nucleotide has deoxyribose sugar. ATP has 3 phosphate groups, whereas DNA nucleotide has only 1 phosphate group. And finally, ATP, the nitrogenous base, is always adenine, whereas in DNA nucleotide, the base can be adenine, thymine, cytosine, or guanine. This is actually where the ATP molecule gets its name from, adenosine triphosphate. It contains adenine and it has 3 phosphate groups. Do make sure with this question that you don't just give one side of the argument. You need to make sure that you're contrasting the 2 molecules. So you wouldn't have got any marks for this question if you just stated what DNA nucleotides have and not compared that to what ATP molecule has. Okay, so just be careful there. So here we're asked to describe how you would use cell fractionation techniques to obtain a sample of chloroplasts from leaf tissue. Now hopefully you'll remember that we actually did this in class using spinach leaves. And don't be deceived by the number of lines here. Sometimes students feel the need to fill up all the lines that are available, but actually you can achieve these 3 marks without saying a lot. The first thing we're going to need to do is break open the cells within the leaf tissue, and this is going to release all of the organelles. And this diagram here represents that quite nicely. So now that we've broken up the cells and released the organelles, we're going to centrifuge our sample. This means that we're going to spin it at a high speed so that the organelles separate out depending on their density. The first pellet you obtain will contain the nuclei of the cells, and this is because the nuclei is the most dense organelle. Then what you're going to do is pour the supernatant out of the test tube, that's the liquid part in the test tube, into a clean test tube, and then centrifuge that again at a higher speed. The chloroplast is the second most dense organelle in a plant cell and will hence form the second pellet. This question here shows us a table outlining some potential features of a mitochondrion and a chloroplast. And we're asked to complete table 3 with ticks where a feature is present. So this is thinking back way to the beginning of your AS year. To help us with answering this question, I'm going to insert a simplified diagram of a mitochondrion and of a chloroplast. If necessary, pause the video now and just review the structure of these 2 organelles. The first feature in the table is a double outer membrane, and we can see from these diagrams that both a mitochondrion and a chloroplast has a double outer membrane. You'll learn more about these double outer membranes in year 2. Other organelles with a double outer membrane include the endoplasmic reticulum, the Golgi apparatus, and of course the nucleus. The next feature is starch grains. So just to recap, starch is an energy storage polysaccharide found in plant cells. Starch is made up of the monomer glucose, specifically alpha glucose, and alpha glucose is produced during the process of photosynthesis. So glucose is a product of photosynthesis. So we would expect to find these starch grains in the chloroplast because this is where photosynthesis takes place. And finally, the final feature is the diffusion of oxygen into the organelle. The mitochondrion is the site of aerobic respiration. So it converts oxygen and glucose to carbon dioxide, water and ATP. So you would expect to find oxygen diffusing into a mitochondrion. Conversely, a chloroplast is the site of photosynthesis. So you would not expect oxygen to diffuse into the organelle. You would in fact expect it to do the opposite and diffuse out of the organelle. Again, going right back to the start of your AS year, the function of a mitochondrion is to produce ATP through aerobic respiration. I've highlighted produce ATP in red and aerobic respiration in blue because actually both of these are worth a mark in their own right. So on to a slightly different question now, a data analysis or graphical interpretation question. Scientists investigated the effect of an exercise program on the number and size of mitochondria in skeletal muscle, so the cells making up the muscles which control the movement of our skeleton. They took samples of muscle from a large number of volunteers before and after the exercise program. From each sample, they cut thin sections and used these to determine the mean number of mitochondria per micrometer squared and the mean area of inner mitochondrial membranes. So they were investigating whether exercise affected the number of mitochondria in muscle cells and also the size of the mitochondria in the muscle cells. And we are asked to explain what the data in Figure 7 and Figure 8 suggest about the effect of the exercise program on mitochondria. So let's look at Figure 7 first. This is comparing the mean number of mitochondria per micrometer squared before training and after training. So was there a difference in the number of mitochondria in the cells before training compared to after training? And the answer is no. And I can draw this conclusion by looking at the standard deviation around the mean both before training and after training. So yes, the mean number of mitochondria per micrometer squared is lower after training than before training, as represented by the top of the bars here shown in red. However, the standard deviation bars here shown in green overlap, meaning that there is no significant difference in the mean number of mitochondria before and after training. So let's have a look at Figure 8 now, which is looking at the size of the mitochondria before and after training. And we can see that the mean, here shown in red, is quite different before and after training. But importantly, we need to consider the standard deviation. And here we can see in green that the standard error bars, these standard deviation bars, do not overlap. There is no value on the y-axis common to the error bar before training and the error bar after training. So there is a significant difference in the mean area of inner mitochondrial membranes before and after training. So we can conclude that training led to an increase in the area of the mitochondria. Just to make sure you're happy with what I mean by the standard deviation bars are overlapping and they are therefore not significantly different from one another, or there are not overlapping, i.e. they are significantly different from one another. I've just inserted these two images here. So the graph on the left, you can see that the standard deviation bars overlap. So there will be values on the y-axis which fall into this standard deviation bar for the first bar and the second bar, okay? So these two means, if it was a comparison, would not be significantly different from one another. The graph on the right, on the other hand, though, we have no overlap. You can see that the error bars themselves do not overlap with one another at all. There's going to be no value that falls in this standard deviation bar, which also falls in this standard deviation bar. That means that there's no overlap and the two means are significantly different from one another. This question here relates to a required practical that we've done in class where we investigated the water potential of potato tuber cells. But question 1 is just asking us to provide 3 properties of water that are important in biology. There are numerous responses that you could provide here, and the nice thing about this question is that no explanation as to why this property of water is important is required. So the fact that water is a solvent would be one response. This simply means that water is capable of dissolving a variety of different substances, for example sodium ions or chloride ions. A second credit-worthy response could be that water is a metabolite. This simply means that it's involved in lots of different reactions, including hydrolysis reactions, where a bond is broken by water being added, condensation reactions, where a bond is formed by removing a molecule of water, as well as reactions such as photosynthesis, digestion, and aerobic respiration. So water is a really important metabolite. A third answer here as to a property of water that is important in biology could be that it has a high heat capacity. This means that lots of energy is required in order to increase a body of water in temperature by 1 degree Celsius. And this is important in biology for a number of reasons. It's important for marine life, meaning that bodies of water don't dramatically change temperature over short periods of time, but also important in the body because it means that a relatively stable body temperature can be maintained. Another property of water that is important in biology is that it has a large latent heat of vaporization. When water changes phase from liquid to a gaseous form, this requires energy, and this is called the latent heat of vaporization. And water requires a lot of energy for it to change state from liquid to gas, hence a large latent heat of vaporization. This property is important for processes such as sweating in order to cool the body. A final response that you could provide to this question is that water is cohesive, or water is held together by hydrogen bonds. Now, we haven't covered this so far in AS biology, but we will do when we have a look at transport in plants. Find this video on YouTube by the Amoeba sisters called The Properties of Water if you require a further explanation of the properties of water. It's a really nice video. Okay, so now on to the practical that this student carried out, which is very, very similar indeed to the practical that we carried out in class. So you'll be able to use your lab books to revise this practical method. A student investigated the effect of different concentrations of sucrose solution on chips cut from a potato. Each chip had the same dimensions. The student weighed each chip at the start, then placed each chip in a separate test tube, each containing 10 centimetres cubed of sucrose solution at a different concentration. So we can see from the table here that they used a concentration of sucrose of 0 moles all the way through to 1 mole. They left the chips in the sucrose solution for 24 hours and then dried the surface of the chips and re-weighed them. And this table here shows their results. Just like we did in class, the student produced the sucrose solutions with different concentrations from a concentrated sucrose solution. So all this means is that their teacher will have provided them with a 1 mole solution of sucrose solution and distilled water. So what they need to do was figure out how much distilled water to put in each test tube, how much sucrose solution to put in each test tube in order to make up the correct concentration of sucrose solution. So for an example of this, if they wanted to make a 0.5 concentration of sucrose solution and they had distilled water and a 1 mole concentration of sucrose solution and they wanted to make up 10 centimetres cubed, they would take 5 centimetres cubed of the sucrose solution at 1 mole and dilute that with 5 centimetres cubed of distilled water. That would give them a total concentration of 0.5 moles per decimetre cubed. So we've done that in class. But we're just asked to name the method she would have used to produce these sucrose solutions, and we call this type of dilution, a dilution series, starting off with a particular concentration and diluting it down to give us a series of concentrations. For the next part of the question, we're asked to use the student's raw data to calculate the ratio of final mass to initial mass of potato chips and plot a suitable graph of our processed data. So processed data is just using the raw data and performing some sort of a calculation. And we're asked to express the ratios as a single number. So the calculation that we're going to do in order to calculate the ratio of final mass to initial mass is simply divide at each concentration of sucrose the final mass by the initial mass. So all we need to do is divide the final mass of the chip by the initial mass of the chip, and that will give us the ratio of the final mass to initial mass of chips. A ratio of final mass to initial mass of chips of 1.36 essentially means that the chip has got 36% larger. Pause the video now and complete the table. So here is what your table should look like completed. Please note that I have stuck to 2 decimal places because that is what has been provided to me in the question. So you should always stick to the number of decimal places provided in the question. Equally, you'll notice that from 0.4 concentration of sucrose, the ratio of final mass to initial mass of chips is less than 1. So what that actually means is that the final mass of the chip was lower than the initial mass of chip, i.e. it has lost mass. A ratio of final mass to initial mass of chips of bang on 1 would mean that the chip had not gained mass or lost mass. It stayed exactly the same. Okay, so let's plot this data. So what is going to go where? Our independent variable is always in the far left-hand column of a table and equally always goes on the x-axis of our graph. It is the variable that we are changing. Don't forget your units when labelling your axes. The independent variable, or the variable that we are measuring, in this case, the ratio of final mass to initial mass of chips, always goes on the y-axis. So this is what we've been measuring throughout our experiment, or the result of processing our data. Ratio does not have a unit. So now we need to come up with a suitable scale for both our x-axis and our y-axis. The x-axis is relatively easy to do this for, and we'll just use each large square as each concentration of sucrose solution provided in the table. As long as your scale on your y-axis is linear and you've tried to make it as big as you can, then you should be absolutely fine. Remember, it's going to be much easier to plot your data if your axes are nice and long. Okay, so now to plot the data. Your curve should look like this. Note that I have joined my data points dot to dot with a ruler, as we do most of the time in biology, and I have not extrapolated beyond the data that was collected by this student. The final part of this question asks us to explain the result for the chip in 0.8 mole per decimetre cubed sucrose solution. You could either use your graph to answer this question at this point here, or you could use the table to answer this question. This question requires an understanding of osmosis and water potential in order to achieve the 2 marks. Now, at 0.8 concentration of sucrose solution, the ratio of final mass to initial mass of chips is 0.81. This means that it has decreased in mass due to the fact that it has lost water by osmosis. The second mark comes from explaining why this has happened. Why has water moved out of the potato cells into the solution? And we know that osmosis is the movement of water from an area of high water potential to an area of low water potential. So if the potato has lost mass, then it must have had a higher water potential than the surrounding solution. We could describe the solution as hypertonic to the cells. Now, this wasn't a question on your mock examination paper, however, quite easily could have been. So it's some good additional research, and it's something that we did in class during this practical, where we used our graph to figure out the sucrose solution concentration, which was isotonic to the potato pieces, i.e. at what sucrose concentration did the potato piece not gain mass or lose mass by osmosis. So just pause the video now and have a little think. How would we obtain that information from this graph? And here is the answer. The concentration of sucrose solution, which was isotonic to the potato pieces, would result in a ratio of final mass to initial mass of chips of 1. That would mean that it did not gain mass or lose mass. So if we go to 1 on our y-axis, we can work backwards to figure out what concentration of sucrose solution is isotonic to the potato pieces. And for this particular question, it is a concentration of 0.33 moles per decimetre cubed. The cells of beetroot contain a red pigment. A student investigated the effect of temperature on the loss of red pigment from beetroot, exactly the same as we did in class. He put discs cut from beetroot into tubes containing water and maintained each tube at a different temperature, and after 25 minutes he measured the percentage of light passing through the water in each tube. Again, exactly the same as what we have done in class. We're told that the student put the same volume of water into each test tube, and we're asked to explain why it is important that he controlled this experimental variable. Now, the first mark comes from saying so that the results from the different temperatures are comparable. If we didn't give each tube the exact same treatment, we wouldn't then be able to compare our different treatments and draw a conclusion. So let's think about why we wouldn't be able to compare if we didn't use the same volume of water. And let's imagine that he used 1 2mm piece of beetroot in each test tube. If at one temperature we used lots and lots of water, and at another temperature we didn't use a lot of water at all, we wouldn't know whether it was the effect of the temperature, which was giving us our absorbance reading, or whether it was just the fact that in the tube with lots of water, the pigment was more diluted than in the test tube with less water. So we need to ensure that the same volume of water was used in each test tube so that we could compare our results at different temperatures. And we knew that it was the temperature which was changing the absorbance of the solution and not simply the dilution of the pigment. For the next part of the question, we're asked to describe a method the student could have used to monitor the temperature of the water in each test tube. Now, the key term here is monitor the temperature, so it implies that he needs to keep an eye on it throughout. So what the student could do is take readings throughout the experiment using a thermometer, so he can continually check that his test tubes are at the correct temperature. Okay, so this question here asks us to draw a suitable curve on figure 2. So this is the graph which corresponds to the previous part of the question. Now don't be fooled by the fact that it says to draw a suitable curve. In biology, and like with all of your required practical graphs, we need to take our ruler and we need to join our data points dot to dot. In biology, we only use a curve of best fit if we can be absolutely certain what intermediate values are going to be. So for example, if this student had a test tube at every 1 degree between 20 and 80. For the next part of this question, we are told that the decrease in the percentage of light passing through the water between 25 degrees Celsius and 60 degrees Celsius is caused by the release of the red pigment from cells of the beetroot. And we're asked to suggest how the increase in temperature of the water caused the release of the red pigment. Here is a section of a plasma membrane. Now, because this is a plant cell, a beetroot cell, there should also be a cell wall, but for the purposes of this explanation, I'm just showing you a plasma membrane. So you can see the phospholipid bilayer, you can see the cholesterol there in yellow embedded in the plasma membrane to regulate fluidity. You can see proteins embedded there, so some transport proteins, some channel proteins. We can also see some glycoproteins, which we know are involved in the immune response and cell recognition. Here highlighted in yellow is the cytoplasm, so inside the cell, and this is where you would expect to see the red pigment. As we increase the temperature of the water above 25 degrees Celsius, we can see that there is a lot more pigment leaving the cell. And this is because at these higher temperatures, we have damaged the cell membrane. And this is because we're increasing the kinetic energy, we are breaking the bonds in these proteins embedded in the plasma membrane, meaning that the proteins with their specific tertiary structure are going to denature. That means that all of this pigment is able to leave the plasma membrane and enter the water, meaning that the percentage of light passing through the water is going to decrease. So here is what you would expect your results to look like. The far left test tube, you can see that not a lot of the pigment has leaked out of the beetroot cells, so this represents a low temperature where the plasma membrane has not been damaged. However, as we move across to these higher temperatures, you can see that the solution is darker, meaning that more pigment has leaked out of the cells because the plasma membrane has been damaged, the proteins embedded in the membrane have denatured, meaning that the pigment can leak out of the cells. Not relevant to this question, but certainly very valuable revision, easily could have been a question. You can see between 60 and 80 degrees on the graph, the percentage of light passing through the water remains constant. And this is because at 60 degrees, the cell plasma membranes have been damaged maximally, so they can't be broken down or destroyed any further. Their proteins have been denatured, the pigment has leaked out, so increasing the temperature further isn't going to further damage the membrane. All of the pigment within the cells has leaked out into the solution. Scientists investigated how the concentration of protein in blood plasma changes in people between the ages of 60 and 95. And figure 4 shows us the scientists' results with standard deviation. If you're not sure how to interpret standard deviation bars, make sure you check out the section of this video relating to interpreting these standard deviation bars. It's on the question about mitochondria before and after exercise. For the first part of this question, we are asked, what is the difference between males and females in the fall in mean concentration of protein in blood plasma between 60 and 95 years? So essentially what this question is asking us is, how much does the male mean concentration of protein in blood plasma decrease between 60 and 95? How much does the female mean concentration of protein in blood plasma decrease between 60 and 95? And then what is the difference between those two? So at 60, the mean concentration of protein in blood plasma for females is 71. At the age of 95, female concentration of protein in blood plasma has dropped to 60 grams per decimetre cubed. This is a decrease of 11 grams per decimetre cubed. So just doing exactly the same for males now. At the age of 60, the mean concentration of protein in blood plasma for males is 69 grams per decimetre cubed. And at the age of 95, it has dropped to 64 grams per decimetre cubed. This is a decrease of 5 grams per decimetre cubed. The question asks us what is the difference between males and females in the decrease, and the answer is 6 grams per decimetre cubed. I got this value by simply taking 5 away from 11. The next part of this question asks us to use the figure provided to calculate the rate of change of the mean concentration of protein in the blood plasma of males between the ages of 60 and 95. So how quickly does the mean concentration of protein in blood plasma decrease for males? And helpfully, we've actually been given the units here of grams per decimetre cubed per year. So between the ages of 60 and 95, the mean concentration of protein in the blood plasma for males decreased by 5 grams per decimetre cubed, as we worked out in the previous part of the question. And this happened over 35 years. So in order to figure out the rate of change in grams per decimetre cubed per year, all we need to do is say, okay, so it changed by 5 grams per decimetre cubed over 35 years, and that tells us that the rate of change for males was 0.14 grams per decimetre cubed per year. As an additional piece of revision, pause the video now and calculate the rate of change of the mean concentration of protein in the blood plasma of females between the ages of 60 and 95. The calculation you will have needed to have performed was 11 grams per decimetre cubed divided by 35 years, and that would give you a rate of 0.31 grams per decimetre cubed per year. This question asks us to use figure 4 to draw a conclusion about the effect of ageing on the mean concentration of protein in the blood plasma in males and females, and this is a 2-mark question. The first mark comes from stating that the protein content decreases with age, and for making the conclusion that it decreases more in females. Thinking back to the previous question that we answered using this figure, we know that the mean concentration of protein in the blood plasma for females decreased by 11 grams per decimetre cubed between the ages of 60 and 95, whereas for males it only decreased by 5 grams per decimetre cubed between those ages. So that's your first mark. Now, if you haven't already checked out the section of this video which explains how to interpret standard deviation, then you're going to need to check that out now so that you understand where the second mark comes from. The second mark comes back to whether or not the standard deviation bars overlap one another, and therefore whether there is a significant difference in the mean concentration of protein in blood plasma between males and females. So if you have a look along the x-axis of figure 4, we have the ages. And you can see at age 60, 65, 70, 75, 80, 85 and 90, the male and female standard deviation bars overlap. So for these ages, there is not a significant difference in the mean concentration of protein in blood plasma for males and females. However, at the age of 95, highlighted here in yellow, we can see that the standard deviation bars do not overlap. So this is your second mark for noticing and making the statement that the difference between the sexes is only significant at 95 years because the standard deviations do not overlap. We're told that scientists measured the absorption of each sample of blood plasma using a colorimeter. They used a calibration curve to find the concentration of protein in samples of blood plasma. So we're asked to describe how the scientists could obtain data to produce a calibration curve and how they would use the calibration curve to find the concentration of protein in a sample of blood plasma. You can find information about how to produce a calibration curve in your biology lab book because we actually did this during the beetroot practical. Now the idea here is that we are trying to determine the concentration of protein in samples of blood plasma by using a colorimeter. However, we need to produce a calibration curve so that we have something to compare to. If we were to take one blood plasma sample and get an absorbance reading of 3 arbitrary units, that's great, but what does that actually mean in terms of protein concentration? So that's where our calibration curve comes in. So what we're going to need to do to start off is to produce known concentrations of protein. And with these known concentrations of protein, we're going to use the colorimeter to measure the absorbance of each concentration. Now this calibration curve is one that I've just taken from Google, so don't dwell on the figures too much, but the idea here is that we've made up known concentrations of protein and then used the colorimeter to give us their absorbance. And by producing a graph like this with our independent variable absorbance on the y-axis and our dependent variable on the x-axis, our protein concentration, and drawing this curve, we now have something to compare our unknown blood plasma samples against. So again, the values that I'm using here are completely hypothetical, but let's imagine that we're working in a lab and we receive a sample of blood plasma from a 75-year-old female, and we need to use this calibration curve to figure out the concentration of protein in this particular sample. So what we're going to do is measure the absorbance of the sample, and let's imagine, again completely hypothetical, that we got an absorbance of 1 arbitrary unit. So that is represented by this red line that I've drawn onto the graph. And then if we just read off the x-axis at the point where the curve reads 1 arbitrary unit, we can see that the protein concentration of this sample is just over 100 micrograms per milliliter. So we've used our calibration curve to determine the protein concentration of an unknown plasma sample. For some additional revision, and if you're not quite sure how to use this calibration curve, pause the video now and work out the protein concentration of an unknown blood plasma sample with an absorbance reading of 0.4 arbitrary units. So what you should have done is had a look at the y-axis and seen that the absorbance was 0.4 arbitrary units for our unknown blood plasma sample. Drawn a line across to the curve and then read off the x-axis the protein concentration which corresponds with that absorbance. So your answer should have been around 20 micrograms per milliliter. The final part of this question tells us that older people are more likely to suffer from infectious diseases. We're asked to suggest how this may be linked to the decrease in the mean concentration of protein in the blood as people get older. Now, because this is a question about disease and infection and illness, there really is only one type of protein that this question could be asking about, and that is, of course, antibodies. As we know, antibodies are large Y-shaped proteins produced by plasma cells which help defend the body against foreign pathogens, owing to the fact that they have antigen binding sites which will bind to the pathogen and help destroy it. Here is a label diagram of the generalised structure of an antibody for some additional revision. We're told that NMO is a disease that leads to damage to nerve cells in the spinal cord. A person with NMO produces anti-AQP4 antibody that attacks only these nerve cells. We're asked to explain why the anti-AQP4 antibody can only damage nerve cells. An antibody is a Y-shaped protein produced by plasma cells. At the top we have the antigen binding sites in the variable region, which is different from one antibody to the next, and we also have a constant region which is the same for every antibody. The variable region of an antibody has a specific structure which is complementary to only one antigen. And in this case, the antigen, the NMO antigen, is only found on nerve cells as shown here in blue. If the specific antigen, complementary to the anti-AQP4 antibody, is only found on nerve cells, then an antigen-antibody complex can only form with these nerve cells, causing the damage. You need to be careful here because the examiner expects you to make specific reference to the nerve cells and the fact that the nerve cells are the ones presenting the antigen, which is complementary to the antibody. Without mentioning the nerve cells, you can only get 2 marks for this question. Scientists measured the concentration of anti-AQP4 antibody in the blood of people with NMO. The spinal cord is surrounded by small bones called vertebrae. For each person, the scientists also determined the number of vertebrae surrounding damaged nerve cells, and their results are shown here in figure 7. A scientist looked at this data and suggested that the concentration of anti-AQP4 antibody in a person's blood could be used to predict the number of vertebrae surrounding damaged nerve cells they are likely to have. So our job is to have a look at figure 7 and suggest reasons why this suggestion might not be valid. First and foremost, if we have a look at figure 7, each dot represents a different individual that partook in this experiment. There's only 20 individuals, and therefore the sample size is too small in order to draw any valid conclusions about the relationship between the concentration of anti-AQP4 antibody and the number of vertebrae surrounding damaged nerve cells. Looking at figure 7, for some of the concentrations of antibody, there is a range in the number of vertebrae surrounding the damaged nerve cells. So for example, highlighted here in yellow, this graph suggests that for one given concentration of anti-AQP4 antibody, there were in fact 3, 4, or 5 vertebrae surrounding the damaged nerve cells. This variation suggests that there is a weak correlation between antibody concentration and the number of vertebrae surrounding damaged nerve cells. In scientific investigations, such as this one here, in order to draw valid conclusions from our data, we need to perform an appropriate statistical test in order to prove mathematically whether two variables, such as the concentration of anti-AQP4 antibody and the number of vertebrae surrounding damaged nerve cells, are statistically correlated to one another. So without a statistical test being used, the scientist cannot draw any valid conclusions from this data. For 7.3, we're told that a new treatment for NMO involves using a monoclonal antibody. The structure of the variable region of this monoclonal antibody is identical to the variable region of an anti-AQP4 antibody, but the rest of its structure is different. We're asked to use this information and our knowledge of antigen-antibody complexes to suggest how this monoclonal antibody prevents anti-AQP4 from damaging nerve cells. So here we have our nerve cell with the NMO antigen displayed on their plasma membrane. Here is the anti-AQP4 antibody, which causes harm to nerve cells. And here is the monoclonal antibody used to treat NMO. Importantly, the structure of the variable region of these antibodies are identical. The variable region is shown in yellow. The rest of the antibody structure is different between the anti-AQP4 antibody and the monoclonal antibody, as represented by the colours purple and white. What this means is that the monoclonal antibody is able to bind to the antigens on the nerve cell, forming an antigen-antibody complex. Because the monoclonal antibody has bound to the antigen on the nerve cells, that means that the anti-AQP4 antibody cannot bind. And in addition to this, when the monoclonal antibody binds to the antigen on the nerve cells, it doesn't cause any damage to the nerve cell. So this is how monoclonal antibodies can be used to treat NMO. Question 10 now about AZT and HIV. I suggest you pause the video now just to have a read of the passage and remind yourself what information was provided. The first part of this question asks us to suggest and explain why AZT does not destroy HIV in the body, but stops or slows the development of AIDS. The important information in the text here is that AZT inhibits the enzyme that synthesizes DNA from HIV RNA. This diagram here is taken from your CGP biology textbook and shows how HIV replicates inside of T helper cells. At point 1, the attachment protein on the HIV viral envelope attaches to a receptor molecule on the cell membrane of the host helper T cell. At point 2, the viral capsid is released into the cell where it uncoats and releases its genetic material, RNA, into the cell's cytoplasm. At point 3, reverse transcriptase, an enzyme, is used to make a complementary strand of DNA from the viral RNA template. At point 4, this double-stranded DNA is made and inserted into the human DNA. At point 5, host cell enzymes are then used to make viral proteins from the viral DNA found within the human DNA. So this is protein synthesis. And then at point 6, the viral proteins are assembled into new viruses which bud from the cell and go on to infect other cells. So that is how HIV usually replicates within a T helper cell. However, we're told that AZT inhibits the enzyme that synthesizes DNA from HIV RNA. If AZT inhibits reverse transcriptase, then HIV particles cannot form new HIV DNA from RNA. So it slows or stops the replication of HIV. So point 3 here is prevented. What this means is the viral DNA is not inserted into the host DNA, no viral proteins are made, no HIV particles can bud out of the helper T cell. If less HIV particles are budding out of the helper T cells, then there will be less HIV particles in the body, meaning less T helper cells will be destroyed and the immune system can continue to work, meaning that it takes longer for AIDS to develop. For the second part of this question, we are asked to suggest and explain 2 advantages of using HAART. HAART involves taking AZT in combination with other anti-HIV drugs. A common error that students make with comprehension questions like this one is they just regurgitate back to the examiner the information provided in the text. What you need to do is use the information provided in the question and then interweave that with your own knowledge and understanding of the topic that you've been taught in class. So one key advantage is for individuals who have become resistant to AZT. So there will be individuals who are HIV positive and for whom AZT is not effective on its own. However, by making HAART therapy available to these individuals, we can slow or stop the development of AIDS because the other drugs provided to the individual will destroy or damage the HIV particles which have become resistant previously. A second advantage is that AZT continues to work as a drug when used in combination with other drugs. This means that any AZT-resistant HIV are prevented from spreading to other individuals. Don't worry if you didn't score very highly on this question in particular. It's a very tough comprehension question. The important thing here is that you start to learn how to answer comprehension questions and how to pick out the important information from the text provided in order to provide credit-worthy responses. So practice makes perfect. At the beginning of the question, we were told that AZT is taken in low doses because people who took high doses over long periods of time suffered muscle wastage. It was found that high doses of AZT inhibit replication of mitochondria. Here we're asked to suggest why high doses of AZT led to muscle wastage. Mitochondria are found in cells and they perform aerobic respiration whereby glucose and oxygen is converted to carbon dioxide, water and ATP. If mitochondria are not replicated and produced in cells, there will be fewer mitochondria and therefore less aerobic respiration. Less aerobic respiration means less ATP and therefore the muscles will waste. Here we're asked to describe how mRNA is produced in a plant cell. Don't let the fact that it says in a plant cell throw you off, because mRNA is produced exactly the same in all eukaryotic cells. We could reword this question and instead say describe transcription, the first stage of protein synthesis. Transcription starts with DNA in the nucleus. DNA is made up of genes, which are sections of DNA which code for a particular polypeptide. So the first thing that needs to happen is that DNA needs to unwind at the particular gene that we are synthesizing the protein for. So the DNA strands separate by the hydrogen bonds between the nitrogenous bases being broken. One of the strands becomes what we call a template strand, and this is what is used as a template for producing our mRNA. RNA nucleotides will use complementary base pairing to form hydrogen bonds with the exposed DNA bases, as you can see here in red. So A on RNA is complementary to T on DNA, U on RNA is complementary to A on DNA, G on RNA is complementary to C on DNA, and C on RNA is complementary to G on the DNA template strand. The enzyme RNA polymerase is needed here in order to form the phosphodiester bonds in the growing mRNA strand. So it's building up the sugar phosphate backbone of our mRNA strand. This process forms what we call pre-mRNA. This pre-mRNA is complementary to the template strand of DNA. But the reason that it's called pre-mRNA is that it contains information or sections which are complementary to introns. Introns are non-coding sections of DNA. So before this mRNA can leave the nucleus and begin translation, we have to remove the introns through a process called splicing. Once splicing has occurred, we have mRNA or messenger RNA, which then leaves the nucleus via a nuclear pore. And that is the end of transcription.